this video, we're going to cover protein structure, function, and regulation. This is a beautiful lecture because proteins are the cell's building blocks and they execute majority of the cell's functions. There are different types of proteins that have different and specific functions. We have enzymes that catalyze many chemical reactions without being consumed by the reaction, structural proteins which provide mechanical support to cells and tissues, transport proteins carry molecules or ions, motor proteins which generate movement. We have storage proteins, transcription regulators, and receptor proteins which detect signals and transmits them to the cell's response machinery. There are also other specialized proteins that act as antibodies, toxins, and hormones. Now, to understand how our bodies function, how genes work, how our muscles contract, we must first understand how proteins operate. So let's get started. Proteins are polymers of amino acids. A polymer is a long molecule consisting of many similar monomers that are linked together by covalent bonds. Recall what covalent bonds are: a strong chemical bond in which one or more pairs of valence electrons are shared. So let's first break down the structure of an amino acid and go through how they are linked. A protein's 3D shape is determined by the location of each amino acid of a polypeptide. And by understanding the structure of a protein, we can see how their specific shape determines its function in a cell. All right? And there are 20 different amino acids in proteins that are coded directly in an organism's DNA, each with different chemical properties. An amino acid is an organic molecule because it contains a carbon. The center of carbon here is called an alpha carbon and it's asymmetric. It has an amino group and a carboxyl group. Another component it has is a side chain, represented here by R. The R group differs with each amino acid. The components of the side chain determines the properties of a particular amino acid, which will influence its functional role in a polypeptide. We can refer to proteins as polypeptides or polypeptide chains. So a protein is made up of amino acids linked together by covalent peptide bonds. Let's go through this. So we have one amino acid here, and let's add another one. A covalent peptide bond forms when the carbon atom of the carboxyl group of one amino acid shares electrons with the nitrogen atom from the amino group of a second amino acid. And we're going to remove a water molecule here. Because we've removed a water molecule, this is known as a condensation reaction. All right, now let's add two more amino acids. Okay, here's our peptide bonds. The carboxyl group of one amino acid shares electrons with the nitrogen atom from the amino group of the second amino acid. Now, the two ends of a polypeptide chain are chemically different. The end carrying the free amino group, NH3+, or it's also written as NH2, is the amino terminus or N-terminus. And then we have this end here carrying the free carboxyl group. And this is the carboxyl terminus or C-terminus. The amino acid sequence of a protein is always presented in the N to C direction, reading from left to right. Now, these amino acids form a polypeptide backbone of repeating structure. This right here, and we have the side chains projecting out. The side chains are not involved in forming peptide bonds, okay? And it's the sequence of these side chains which give each protein its unique properties. Some of these side chains are non-polar, they don't have a charge, and hydrophobic or water-fearing, while others are positively or negatively charged. Now, I mentioned earlier that by understanding the structure of a protein, we can see how the specific shape of a protein determines its function in a cell. There are 4 levels of organization in the structure of a protein, known as primary, secondary, tertiary, and quaternary structure. Quaternary structure occurs when 2 or more polypeptide chains combine, okay? So let's now subtract complexity and go through the primary structure. The amino acid sequence is known as the primary structure, all right? The primary structure is the sequence of amino acids. Let's actually show the atoms here. Oh, gorgeous. This is one amino acid. the carboxyl group is bonded to the nitrogen atom of this amino acid, and we have our side chains projecting out. The sequence of amino acids is determined by inherited genetic information. It's not just random combinations of amino acids. For example, one molecule of human insulin should have the same amino acid sequence as every other molecule of human insulin. The primary structure then determines the secondary structure, which involves alpha helices and beta pleated sheets, as well as the tertiary structure. And this is due to the backbone and the R groups, or side chains of amino acids along the polypeptide. So from primary structure, the next level is the secondary structure. The secondary structure involves coiled or folded patterns in the polypeptide chains known as alpha helices and beta pleated sheets. How does this occur? Let's go through this. These coils and folded patterns result from the hydrogen bonding between the atoms in the polypeptide backbone without involving the side chains, okay? So within the backbone, the oxygen atoms have a partial negative charge. They're electronegative. And the hydrogen atoms attached to the nitrogens have a partial positive charge. Polar molecules like polar molecules, and so these hydrogen bonds can form hydrogen bonds between these atoms. If you recall, hydrogen bonds are weak, but because there's so many of them, they can support a particular shape for that part of the protein. And there are two types of secondary structure, which were both discovered from studies of hair and silk. The first pattern is called the alpha helix, which is a coil held together by hydrogen bonding. The hydrogen bonds can be found between every fourth peptide bond, linking the carbonyl group of one peptide bond to the hydrogen atom attached to the nitrogen of another. The second structure is the beta pleated sheet. So a beta sheet is made when hydrogen bonds form between segments of a polypeptide chain that lie side by side, called beta strands. They can form either from neighboring segments of the polypeptide backbone that run in the same orientation, okay, parallel, from the N terminus to the C terminus. And this structure is called a parallel beta sheet. Or they can form a polypeptide backbone that folds back and forth upon itself, with each section of the chain running in the opposite direction, anti-parallel chains. And this structure is called an anti-parallel sheet. Both types produce a very rigid structure, and they form the core of many proteins. Okay, alpha helices and beta sheets are common folding patterns. So that's the secondary structure, which involves interactions between the polypeptide backbone. The next level of organization is called the tertiary structure, which is the overall shape of a polypeptide resulting from interactions between the side chains. So the tertiary structure is the full three-dimensional conformation, the shape of a polypeptide. The folding of a protein is determined by different types of weak non-covalent bonds that form between one part of the chain and another. These involve atoms in the polypeptide backbone as well as atoms in the side chain. There are four types of these weak bonds. We have hydrogen bonds, electrostatic, van der Waals attractions, and a hydrophobic clustering force. If you'd like a refresher on the different types of chemical bonds that form between atoms, watch the molecules and chemical bonds lecture and then just come back to this one. Alright? Now, in individual non-covalent bonds, like we mentioned before, they're extremely weak in comparison to the typical covalent bonds that produce biological molecules. But if we have a lot of these weak bonds forming, they can hold two regions of a polypeptide chain tightly, and they stabilize the tertiary structure. They come together to create a strong bonding arrangement. Teamwork. So these are the different interactions that occur between side chains. Speaking of side chains, recall that side chains can be polar or non-polar, and these properties influence the folding pattern of any protein. Non-polar side chains are hydrophobic, which means they don't like water. So what will happen is they're going to cluster in the interior of the folded protein so that they can avoid contact with water. The non-polar side chains are located on the inside, so they're hidden from water, and they're going to form a tightly packaged hydrophobic core. Recall that non-polar molecules don't have charged regions, so they will not interact readily with polar molecules, whereas polar molecules have charged regions, so they tend to arrange themselves near the outside of the molecule where they can form hydrogen bonds with water and with other polar molecules. These are weak non-covalent bonds, but we can also form covalent bonds called disulfide bridges, and these can form between two cysteine side chains of a protein if they are brought close together. Now, most proteins have a particular three-dimensional structure, which is determined by the order of the amino acids in its chain. And the final folded conformation is generally the one that minimizes free energy. As a result, the folding process is energetically favorable as it releases heat and increases the disorder of the universe. Disorder is a term to describe how spread out the energy is in a system, and a measure of the degree of disorder is called entropy. And so entropy allows us to understand why some processes are more energetically favorable. If you'd like a refresher on these concepts, watch the energy lecture. That's a big one. Going back to proteins, how do we know that the 3D structure of a protein is contained in its amino acid sequence? Protein folding has been studied using highly purified proteins. We can unfold the protein or denature it by treating it with solvents that disrupt the non-covalent bonds that hold the chain together. And under the right conditions, when the denaturing solvent or treatment is removed, the protein often refolds back to its original shape, original conformation spontaneously. And this is a process called renaturation. So this renaturation process tells us that the 3D shape of a protein is specified by its amino acid sequence. Okay, moving on. The weak non-covalent bonds we mentioned also allows proteins to bind to each other to produce larger structures in the cell. Any region on a protein surface that interacts with another molecule via non-covalent bonds is termed a binding site, and a protein can contain binding sites for a variety of molecules. If a binding site recognizes the surface of a second protein, the tight binding of two folded polypeptide chains at this site will create a larger protein. And this is where the quaternary structure comes in, when two or more polypeptide chains combine. Each polypeptide chain is called a subunit, and they may contain more than one domain. Now, a protein chain can fold in its correct shape without outside help, but protein folding in living cells is generally assisted by chaperone proteins or molecular chaperones. These chaperones bind to partly folded chains and help them to fold along the most energetically favorable pathway. So recall that the final folded conformation is generally the one that minimizes free energy. So chaperone proteins make the folding process more efficient. Think of them as guardians that hold the proteins together and help them, assist them in their folding process, their folding journey. Some chaperones can act as isolation chambers that help a polypeptide fold without the risk of forming aggregates in the crowded conditions of the cytoplasm. This requires ATP binding and hydrolysis to put this little cute chamber cap on. Okay, the chaperone proteins assist the folding process. They guide the protein chain to make sure that they are folding efficiently. But what happens? Let's take a look at what happens when proteins fold incorrectly, because this can happen. They can form amyloid structures that can damage cells. Amyloid fibrils are beta sheet aggregates, and they're used for some normal cell functions, but when they are not controlled, they can contribute to a number of neurodegenerative disorders such as Alzheimer's disease and Huntington's disease. Misfolded proteins are called prions. The misfolded prion form of a protein can convert the properly folded version of the protein in an infected brain into the abnormal conformation. Then these abnormal prion proteins grow, and they are going to aggregate to form amyloid fibrils, which can disrupt brain cell function, causing a neurodegenerative disorder. And prion disease can spread from one organism to another via contaminated food or blood, so it's considered infectious. All right, so we've covered a lot so far, and how proteins have several levels of organization: primary, secondary, tertiary, and quaternary. But there's another level of organization called the protein domain, which is any segment of a polypeptide chain that can that can fold independently into a stable structure. Alright, so we've covered how each type of protein consists of a precise sequence of amino acids that allows it to fold up into its unique three-dimensional shape or conformation. We also covered how chaperone proteins assist the folding process and what happens when proteins fold incorrectly. This is beautiful and all, but how do proteins actually work? Let's break down how the activity of proteins depend on their ability to bind specifically to other molecules. We'll go through a few examples, but essentially the specialized functions of proteins are based on similar principles. So the biological properties of proteins depend on its interaction with other molecules. Proteins bind or stick to other molecules. This binding can be very tight or weak. For example, the enzyme hexokinase binds glucose and ATP to catalyze a reaction between them, or antibodies attaching to bacteria or viruses as part of the body's defenses. How does this work? Let's break this down. Proteins are very specific in the molecules they bind with. They're quite picky. Each protein molecule can bind to just one or few molecules. Any substance that is bound by a protein, whether it is an ion, a small organic molecule, or a macromolecule, is referred to as a ligand for that protein, from the Latin ligare, which means to bind. So here we have our ligand and our protein. Recall that the region of a protein that associates with a ligand is known as its binding site, which is formed by a particular arrangement of amino acid side chains. The ability of a protein to bind selectively is due to the formation of weak non-covalent interactions, bringing back the weak non-covalent bonds that help stabilize protein structure. We have hydrogen bonds, electrostatic attractions, van der Waals attractions, and hydrophobic forces. So these red lines are non-covalent bonds. Earlier we mentioned these bonds are weak, but if we have a lot of them forming, it's quite strong, and teamwork applies here. As well as the surface of the ligand needs to fit to that protein, because if the molecules aren't matched closely to the protein, only few non-covalent interactions will form, and the two molecules will dissociate because the binding is not strong enough. This mechanism is what prevents incorrect associations. All proteins must bind to specific ligands to carry out their various functions. Let's call them soulmates. They're meant to be. So let's go through an example using antibodies, because the body is capable of producing billions of different antibodies that recognizes and binds tightly to so many different types of ligands, molecules found on bacteria and viruses. Antibodies are immunoglobulin proteins produced by the immune system in response to foreign molecules, especially those on the surface of an invading microorganism. Each antibody binds to a particular target molecule extremely tightly, either inactivating the target directly or marking it for destruction. An antibody recognizes its target molecule, called an antigen. So, how are a wide variety of different antibodies formed? Let's go through this. It's amazing. Antibodies are Y-shaped molecules. Each molecule is made of two identical light chains and two identical heavy chains. So then its antigen binding sites are also identical. The antigen binding sites are formed from several loops of polypeptide chains. The amino acid sequence in these loops can vary greatly without altering the basic structure of the antibody. And we can form a wide variety of antigen binding sites through changing the length and the amino acid sequence of these loops. All right, let's go through another example. Let's use enzymes this time. Enzymes are an important class of proteins. They act as catalysts, which means they speed up chemical reactions without being consumed by the reaction. Enzymes bind to one or more ligands called substrates, and then the catalytic behavior of the enzyme transforms the substrates to the products, and they can do this over and over again without themselves being changed. We go through this in detail in the enzyme function and regulation lecture. All right, so now that we've covered how proteins bind to other molecules to perform their function, let's now break down how protein activity in the cell is controlled and regulated to maintain an optimal state. Because the cell's job is to coordinate every chemical reaction to make sure it's producing the appropriate amount of substrates and products and it's not wasting energy. There are several levels that occurs to control and regulate protein activity. One mechanism is to control the expression of the gene that encodes the protein. It can regulate the rate at which the protein is degraded, or it can control protein activities by placing the protein in a subcellular compartment, or it can be regulated at the level of the protein, which we'll go through. Now, all these mechanisms still involve interactions with other molecules and other proteins. So let's go through the first mechanism, and that is how enzymes are regulated by other molecules. So like we mentioned before, enzymes are powerful biological catalysts. Their activity depends on their protein conformation, its primary, secondary, tertiary, and quaternary structure. If an enzyme is denatured, their catalytic activity is gone. A common type of control occurs when a molecule other than a substrate specifically binds to an enzyme at a regulatory site, altering the rate at which the enzyme converts its substrate to product. Okay? In feedback inhibition, for example, an enzyme acting early in the reaction pathway is inhibited by a molecule produced later in that pathway. Let's break this down. So in this example here, we have two different pathways that are connected. Substrate B is the first metabolite that gives the end product Z in this pathway here. Substrate Z is going to inhibit the first enzyme that is specific to its own synthesis, and so it's going to limit its own concentration. So if a reaction produces large quantities of the final product, the products are going to accumulate and they're going to bind to an early enzyme, okay? In this case, the first enzyme that catalyzes the reaction from B to X, and it's going to slow down its catalytic reaction, its catalytic action. The end result, this is going to be limiting further entry of substrates into this reaction pathway. Now, feedback inhibition is a form of negative regulation, which means it prevents an enzyme from performing its function. And enzymes can also be subject to positive regulation, in which the enzyme's activity is stimulated by a regulatory molecule rather than being suppressed. Positive regulation occurs when a product in one metabolic pathway stimulates the activity of an enzyme in another pathway. But the question is, how do these molecules, regulatory molecules, change an enzyme's activity? And this is where allosteric enzymes and allosteric regulation comes in. Let's go through this. Most enzymes are allosteric proteins, which means they can exist in two conformations or shape that differ in catalytic activity. So think of it, they have two personalities, all right? And these enzymes can be turned on or off by ligands that bind to a specific regulatory site to stabilize either the active or the inactive form. So allosteric proteins have two personalities, the active form and inactive form. And ligands can bind to specific regulatory site to stabilize either the active personality or the inactive personality. Let's break this down. Most enzymes known to be allosterically regulated are produced from two or more subunits, each composed of a polypeptide chain with its own active site. Allosteric enzymes have two or more binding sites that influence one another, okay? So this here is the active form, okay? Here's an active site and allosteric site. And then we have our inactive form with a non-functional active site. So think they have two personalities. We have the active form and the inactive personality. These active sites communicate with each other in such a way that a shape change in one subunit is transmitted to all of them. The interactions between these active sites depend on a conformational change in the protein. And again, many enzymes have two conformations that differ in activity, each of which can be stabilized by the binding of a different ligand. If an activator binds to a regulatory site, it's going to stabilize the shape that has functional active sites. Whereas if an inhibitor binds, it's going to stabilize the inactive form of the enzyme. Now, this doesn't just apply to enzymes, but it also applies for many other proteins as well. So that's feedback inhibition and allosteric regulation. So think allosteric, they have two different types of personality, okay? The active form and the inactive form. And a ligand combined to a regulatory site, which can stabilize either the active personality or the inactive personality. That's a really great way to remember allosteric. Think allosteric, two personalities. Another mechanism that can control protein activity and cause a conformational change from inactive to active or vice versa is phosphorylation. This involves attaching a phosphate group covalently to one or more of the protein's amino side chains. Phosphorylation can either stimulate protein activity or inhibit it, depending on the protein involved and the site of phosphorylation. And we cover this across the metabolism lectures. It was absolutely beautiful. So if you get to the metabolism lectures, you're going to see phosphorylation occurring a lot. The addition and removal of phosphate groups from specific proteins often occurs in response to signals that specify some change in a cell state. Let's break this down. So we have our protein here in its dephosphorylated form. It doesn't have a phosphate group. Protein phosphorylation involves a transfer of a phosphate group from ATP to an amino acid side chain of the target protein here. Specifically, the phosphate group of ATP is transferred to the hydroxyl group on a serine, threonine, or tyrosine side chain of the protein. The addition of a phosphate group can cause a conformational change, and a structural shift alters the protein's activity. So this protein has changed in structure and we have stimulated its activity. We've turned it on now. Okay? And this reaction is catalyzed by a protein kinase. Now, if we remove the phosphate group by a second enzyme, the protein will go back to its original shape, original conformation, and it can continue performing its initial activity. So the reverse reaction involves dephosphorylation, or the removal of the phosphate group, and this is catalyzed by a protein phosphatase. Okay, so think phosphorylation, we're adding a phosphate group, and dephosphorylation, we're removing that phosphate group. Reversible protein phosphorylation controls the activity of many types of proteins in eukaryotic cells. Now, we can also flip this, okay? So here we have the dephosphorylated form as the active form, and we can phosphorylate it. We can add a phosphate group from ATP. So here's our cute phosphate group attached, and this is catalyzed by a protein kinase. And the addition of a phosphate group inhibits protein activity. We've turned it off now. We're like, hey buddy, here's a phosphate group, chill out, we don't really need you right now. And so to stimulate it again, all we have to do is remove that phosphate group and it'll turn on. Gorgeous process. All right. So these are just a few mechanisms cells use to control and regulate protein activity. In this lecture, we learned that each type of protein has a unique amino acid sequence which determines both its unique three-dimensional structure and biological activity. We covered how the biological function of a protein depends on the specific chemical properties of its surface and how it binds to other molecules called ligands. Lastly, how the activities of proteins are controlled and regulated in a cell through feedback inhibition, allosteric regulation, and phosphorylation. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!